Immune-Leish Symposium. So yesterday we had an amazing time to discuss science and immunology and leishmaniasis, which we love. So today we are going on for our second day to discuss about immunopathogenesis. So I'm going to give the word for our chair. Thank you so much, Cláudia, Dr. Cláudia, Professor Cláudia, to accept to be shared today. So Cláudia is a research fellow from CNPq, 1A, so it's the maximum level in Brazil, and she's researching in FIOCRUZ, Bahia. She's retired from Federal University of Bahia with many contributions in immunopathogenesis for leishmaniasis. Thank you so much, Cláudia, to accept to be chair today.
Hi. Hi, everybody. Good afternoon. Thank you very much for the so kind words, Herbert. It's really a great pleasure to be here today just to announce our speakers in this afternoon about immunopathogenesis and leishmaniasis. So our speakers will be Phillip Scott, or Phillip Scotti. He is a professor of microbiology and immunology, Department of Pathobiology, University of Pennsylvania, School of Veterinary Medicine, Vice Dean for Research and Academic Research, also in the University of Pennsylvania, member immunology graduate group, and cell and molecular biology graduate group. So in his lab, the main research areas are host pathogen interactions, leishmania, memory T cells, immunology, infectious disease, leishmaniasis, and most recently, the Scott Lab has begun studies in Brazil, funded by the NIH International Collaborations in Infectious Disease Research grant to understand the pathogenesis of Leishmania brasiliensis. Phil, it's really a great pleasure to get you here. And I think you can start, and after this, I can introduce the other speakers, Lucas Carvalho and Danielle Walnis. Thank you very much for accepting our invitation.
Okay, so do you see my slides?
Yeah.
So first of all, thanks for the invitation. I'd rather be there than here, but I'm still happy to have the opportunity to talk a little bit about what we're doing. Actually, Cláudia, it's not that recent that we started working in Brazil. When I think about it, really extensive, most of the stuff we've been doing, we started almost 10 years ago. But the first time I went to Brazil and went to Edgar's lab, frighteningly, for some of you to know this, it'll speak to my age, it was in 1983. So a long time ago, and it's a place I really like to go, and I'm hoping to be able to get back there soon. So again, thanks for the invitation. What I'm going to talk about today is some of the work that we've done in Philadelphia and some of the work we've done in Brazil. And mostly the point I really will make on the next slide is how this data that I'm going to present is really a combination of work that's been done by a lot of different people. So I'm going to talk about the immunopathology of cutaneous leishmaniasis. And to make that point, I want to start off with thanking the people that have done this work. I'm going to talk about two aspects of immunopathology. One is CD8 T cell cytotoxicity, and the other is the skin microbiome and how the skin microbiome may be influencing disease and in leishmaniasis. And so multiple people from my lab have been involved. I'm going to point them out as I go through the talk. Dan Biting, who's been critical for a lot of the bioinformatics works that we've done. Elizabeth Grice, who's been critical for all the microbiome work. And particularly Edgar, Lucas, and their colleagues in Brazil. And almost all of the work that we've done has been dependent upon them and the human studies that I'm going to be talking about. Just quickly, this is a Leishmania group, so I'm not going to spend a lot of time talking about leishmaniasis, but just to kind of frame what we've been interested in, a long time we've been interested in what are the factors that are driving pathology in this disease. And I think this is a slide for those of you working on leishmaniasis will be quite familiar with. There's multiple clinical forms of leishmaniasis. And at the left-hand spectrum and left-hand side of this slide, I'll go back. Let me get my laser. On the left-hand side of this slide, you see patients that have diffuse cutaneous leishmaniasis, and these are patients who have large number of parasites, and we can understand that they're not developing an effective immune response, hence they're unable to control the parasites. But as you go across the spectrum, you see patients with localized disease, severe localized disease, and this is a patient with Leishmania brasiliensis, and then the most severe form at this end of the spectrum, mucosal disease. And the interesting immunologic question has been, what promotes this increased pathology, even though for the most part, these patients are able to control the parasites. And so that's been a question that I think lots of people in the field have thought about for a long time. And one of the problems, and I'll mention this in a minute, has been that there haven't been great experimental models in order to understand that pathology. But if we had models and if we understood that pathology, the rationale to study the disease was that we might be able to develop host-directed therapies that could lessen disease severity. And so if you don't have experimental models, what you do is you go to the real deal, you go to patients. And so this slide shows, again, the place where we've collaborated with Edgar and Lucas. This is the Leishmania Reference Center in Corte de Pedra. And they see a large number of patients there. And all of the studies, the human studies that have been done with samples or patients from this site. So in order to understand this disease, if you don't know much, one of the things you want to do is just get a general picture of what is the immune response in these patients. And so I'm going to talk about in this talk, I'm going to go through studies that have been involved in looking at the transcriptional analysis of lesions from patients with Leishmania brasiliensis. And we've had 3 different cohorts that I'm going to be talking about. The first actually is a cohort that we described, we obtained quite a long time ago now, 2012, and started publishing on it. The first paper was in 2013. And the question there was a very simple question: what genes are upregulated in Leishmania brasiliensis? The second cohort, which I'll talk about, we asked a different question, and that is what genes are associated with treatment failure. And finally, I'm going to present a little bit of the unpublished data on a cohort that we're still analyzing, asking whether the skin microbiome influences host gene expression. So in all of these studies, what was done was to collect clinical data and blood from the patients when they came into the clinic before treatment. In the third cohort, we also collected skin swabs and bacterial isolates. And then in all of the cohorts, as I said, we collected biopsies. Again, it's important to mention they were before any treatment. So the first study was actually not an RNA-seq study. It was old school. It was a microarray. And I kind of like the fact it was a microarray because it's quite visual in looking at it. And again, what you see here are genes that are upregulated in red and genes that are downregulated in blue, comparing lesions and normal skin. And these are studies that were initiated by Fernando Novaes, who was a postdoc in the lab, who now is an assistant professor and has her own lab at Ohio State University. And so if you were looking at patients with Leishmania brasiliensis, obviously one would expect high levels of interferon gamma. And in fact, you can see here that in this particular microarray, you see gamma interferon highly upregulated. What we didn't expect, and maybe we should have if we'd paid more attention to Claudia's work, what we didn't expect was such a high upregulation of granzyme B, granulysin, and perforin. And so this made us begin to think about, well, what are these factors playing? What role are they playing in the disease? And And one possibility is that these were upregulated because we thought, well, maybe CTLs are important in controlling the parasite. And so this is a protective mechanism. But as I mentioned earlier, we did have experimental models to begin to ask the question, whether or not they were or they were not protective. And so what Fernanda did was she began to develop some experimental models in mice to ask that question. And the most reductionist model, and the one we've worked a lot with, is a CD8 RAG model, which is quite simple. It's where we transfer into RAG mice CD8 T cells. You get a severe lesion. If you transfer in CD4 and CD8 T cells, you get control. And interestingly, if you infect mice, RAG mice, and don't transfer any cells, you actually don't get much of a lesion. But in fact, the number of parasites are quite high. So the number of parasites between those mice that have CD8 T cells and those that have nothing, really about the same. And so it's really about pathology. We also developed some additional experimental models to look at the pathologic role CD8 T cells can play. And these had to do with having an infection which would lead to high levels of CD8 T cells. And we chose lymphocytic choriomeningitis virus, which we know induces a high number of CD8s. And we asked the question, if you had a previous infection with LCMV and then challenged with leishmania, would you see increased disease? And what you can see in the middle panel here is, in fact, those LCMV immune mice do develop more severe disease. And that more severe disease is not because of CD8s that are specific for leishmania. They're actually specific for LCMV, but they're recruited to the lesions, and in an NKG2D dependent way, they're able to lyse cells in the lesion, leading to pathology and this increased disease. And again, importantly, there's no difference in the parasite numbers. It's simply a matter of increased pathology. And finally, we've played with that model a little bit, and you can do a co-infection with LCMV and get similar kinds of results. And so these are the models that, some of the models that we've been talking about. I'm going to show a slide that summarizes both those CD8 cytolytic models that cause disease, as well as some of the other models we're looking at. One is dysbiosis, which I will be talking about a bit later. But we're also looking at the lack of regulation, and this is an IL-10 deficiency model, as well as looking at a strain that David Sachs has been working on for a long time, Seidman strain, which causes this severe disease as opposed to more healing infections with L major. And the important point is that in all of these models, NLRP3 is critical for the infection and the disease, I should say. IL-1 beta is critical as well. So all of the data that we've done so far, as well as work from Cláudia Brodskyn's lab, has led to this idea that there is a pathway leading from leishmania-specific CD8 T cells to inflammasome activation, IL-1 beta production, leading to inflammation. The systems, there have been different systems of different studies that have been done in patients, studies that have been done in mouse, and it seems pretty robust that this is an important pathway. In addition, as I mentioned, these CD8 T cells don't have to be specific. And so when we think about what might be happening in a situation where individuals have had previous exposures to viruses that may have increased their CD8 repertoire of, or I should say, number of CD8 T cells, one kind might envision that they also could lead to increased pathology in this non-specific way. And this isn't just something that we think might be happening in the patients. Fernanda also was able to show that if she looked at CD8 T cells in Leishmania brasiliensis lesions, they do express this molecule NKG2D, and myeloid cells express the MKG2D ligand MICB. And this is data from patients showing CD8 T cells expressing NKG2D, as well as from lesions, the gene expression of MICB in healthy skin versus skin from Leishmania brasiliensis patients. So this pathway really made us think that this was an important component to the pathology that we were seeing in patients. And the studies from the mice were pretty convincing. And studies from the patients that we did and Claudia Brodskyn did also were pretty convincing. But it really was difficult to know for sure whether in fact these were indeed associated with some outcome in the patients. And so really without a clinical trial, it's kind of difficult to completely conclude that cytolysis and IL-1 beta is promoting increased disease. And so we asked the question whether genes associated with cytolysis and IL-1 beta expressed higher levels in patients that fail antimony treatment. And again, for those that don't know, in the northeast of Brazil, in this particular site, the treatment with antimony is about 21 days of 20 mg per kg given intravenously. And unfortunately, a large percentage, between 40 and 60% of the patients, fail that treatment the first time. After a number of days, actually after 90 days, the patients are reevaluated. And if they fail treatment, they may go on another round of antimony. So that's a pretty severe treatment, but it also allowed us the opportunity to compare gene expression in patients that were going to fail or that we're going to cure. And so again, I'll make the point that these patients come into the clinic, all of the data that we're going to show now is from biopsies that were taken before they were treated. And then retrospectively, we look to see whether or not patients failed treatment or cured after treatment at a 90-day time point. So these studies were done by another postdoc, Camila Amorim from Brazil, and collaborating with Dan Beiting, who's been critical for the analysis of the patients. So, so not surprisingly, we knew when we did this second cohort of patients that the genes that would be highly upregulated would be genes associated with cytolysis as well as IL-1 beta. And you can see this here, granzyme B, perforin, granulysin, and IL-1 beta. You see high levels in the orange color from the patients. Each dot is an individual patient compared to healthy skin. But you can also see pretty clearly that there's a large variation in expression among the patients. And so the fold change from the most highly expressed to the least highly expressed can be over 100-fold difference. And so what this allowed us to do is to begin to think about whether those variable genes might be dictating what happens, whether a patient fails or cures. And so what Camila did was she went back to the data set of these patients, in this case, a data set with 21 patients, and did a coefficient of variation to pick out those patients where the lesions, or I should say where the gene expression was most variable, and looked at, which is shown here in this red circle. And so there were a lot of genes that were in this group. This was about 250 genes. And within those 250 genes, in an unbiased way, granzyme B, perforin, granulysin, and IL-1 beta were there. I will say that a lot of these other genes, and you can see them in yellow here, turned out to be related to B cell responses. And there were a lot of chemokines genes that were in red here. And so having this set of genes, Camila could then ask whether or not they were associated with treatment failure. And so what's shown on this slide is the p-value of failure versus cure of this set of genes versus fold change. And what you can see is that there's a set of genes in this box that are highly expressed in terms of fold change between failure and cure, and also significantly expressed. And these included the genes that we were kind of interested in, because we thought were going to be important in influencing disease: granzyme B, perforin, granulysin, and IL-1 beta. Just because some people might ask, what about all those B cell genes? They're down here in the yellow. And in fact, they don't seem to be involved in driving whether or not a patient's going to resolve the infection or not after treatment with antimony. So the cytolytic IL-1 beta pathologic pathway is definitely associated with treatment failure. As I said, there's other genes that are associated with treatment failure, some of which we're particularly exploring now. And kind of from a useful point of view, this higher expression of cytolytic genes allows us to predict treatment failure. And so last March, Dan Beiting and I had tickets, not last March, March of 2020. We had tickets to leave Philadelphia to go to Brazil on March 16th, and the university shut down on March 16th. And we were going down to Brazil to specifically begin to think about how we could look at patients prior to treatment to decide whether or not they would fail treatment. So that's kind of on hold, but I still think with the data we have, I think it's very possible to be able to do that. The other thing I'll point out, and this is something that we didn't expect, although it's somewhat predictable, because these patients have very few parasites, but the patients that failed treatment had slightly more parasites than the patients that cured. And so at the moment now, there's a group of people, including Dan Beiting and others, who are now looking at whether or not differences in parasites may be dictating what's happening in these lesions as well, in addition to just the host immune response. So having this information, one of the things that Fernanda was particularly interested in was asking the question of, can we see whether or not we can block this pathology with some of the obvious treatments that you would think of? And so an obvious treatment thinking about blocking IL-1 beta would be blocking IL-1 beta with an antibody. And there is a humanized monoclonal antibody against IL-1 beta, canakinumab, which could be used in patients, as well as another drug, anakinra, which has been used for many years, which blocks IL-1 beta. And what's shown on this slide here in the data is a RAG CD8 T cell model where you have this course of infection of mice that have got CD8s. They develop severe pathology, and the picture is helpful here. If you treated if you treated the mouse with anakinra, you see a decrease in the ability of that pathology, and you see that here. And that was true in this RAG CD8 T cell model, as well as in at least an LCMV co-infection model, where if you blocked IL-1 beta with anakinra, you blocked that severe pathology you see here, and it was brought down to the same amount of lesion that you would see without LCMV. And so this is kind of just an example of some of the experiments that were done. Fernanda was also able to go on and show that you could block NLRP3 with a couple things. One was MCC950, as well as glyburide. And the other was a JAK1/3 inhibitor, tofacitinib, which not only worked systemically, but importantly also worked topically. And so at the moment, we're looking at these and mechanisms of action and thinking of other pathways that may be involved in the pathology. And so I'm going to switch now to what other additional targets might be involved in host-directed therapies. And one way in which we're thinking about that is asking, what are the cells that are involved in this inflammasome activation? And so a postdoc in my lab, Christina Go, is looking at the cells, in this case, using a mouse that expresses citrine when the ASC part of the NLRP3 inflammasome is activated. And this is a lesion of a mouse infected with Leishmania major. You see the lesion here, and each of these dots are the aspect that's associated with inflammasome activation. And this is allowing Christina to be able to identify the cells that are actually undergoing inflammasome activation, with the idea that if we knew what those cells were, that would help us in terms of blocking this pathway. Camilla has also just published a paper asking what the systemic changes are in gene expression. And for those that are interested, that's a paper that just came out in PLOS Neglected Tropical Diseases. But again, what other pathways might promote disease? And so a number of years ago, a graduate student in my lab, Sierra Gimlet, who's now a scientist at GSK, started a collaboration with Elizabeth Grice, who works on the skin microbiome, to ask the question whether or not the skin microbiome plays a role in this pathway. And so it was an obvious thing to look at because we know that bacteria in the skin can induce IL-1 beta, and clearly the possibility that they would induce more pathology was something that we thought of. And so this is the results from a paper from a few years ago where Sierra was able to show that if you took mice and you made them dysbiotic, and in this case, by co-culturing, co-housing mice that were infected with Leishmania with naive mice, and showing that they could transfer that dysbiosis in the skin to the mouse that was not infected, and then infecting the mice, these animals developed much more severe disease, both by a pathology score, and actually the picture of the pathology is probably the most useful way to look at it. And importantly, the parasite burdens were similar. This increased severity of disease depended on IL-1 beta, as well as IL-17, and was associated with neutrophil recruitment. This isn't a total surprise. Yasmin Belkade, a number of years ago, showed that germ-free mice develop less inflammation following infection with Leishmania major, suggesting that the skin microbiome is playing a role in pathology in this disease. And so this led to the third kind of set of studies that we're doing, actually ongoing studies right now, where we have, depending on which part of the study, between 50 and 64 samples from patients. And primarily that's what we're studying. We have a few healthy subjects involved, but primarily we're looking at the patient studies where we're collecting skin swabs and bacterial isolates. So in this study, we've done 16S sequencing. We've characterized the bacterial isolates over time, changes over time in terms of the 16S sequencing, as well as are currently characterizing the bacterial isolates that we have. Again, of course, we collected biopsies. We've done RNA-seq on those, qPCR for parasite burden, qPCR for bacteria, as well as selective whole genome amplification of the parasites. And that's work that Dan Beiting's doing right now, actually. And so the three questions that I'm going to answer, and I'll point out again that this is work that's ongoing. So it's still a work in progress, but we have some data that I thought would be useful to show to you that we haven't yet published. So the first question was, does the abundance of bacteria and the lesions influence host gene expression? Here we've done qPCR for the 16S ribosomal gene in lesions and correlated with RNA-seq host gene expression. We've asked what type of dysbiosis do we see in lesions? Again, 16S sequencing of the swabs. And finally, we asked the question, do bacteria from lesions promote increased disease in mouse models? And here, the data I'll show you is associating bacterial isolates with mouse skin and assessing immune responses in the course of infection. So first, just asking a question of, does it matter how much bacteria is there, whatever type of bacteria it is? And so this was a fairly simple experiment to ask, what was the total 16S abundance in healthy skin versus skin from lesions of patients? And what you see here is, not surprisingly, an increase in the abundance of bacteria from lesions from cutaneous leishmaniasis patients. And when one looked at RNA-seq analysis of host gene expression associated with this increase in parasite and bacterial abundance, you saw an upregulation of a lot of genes, including those associated with chemotaxis. So CXCR1, CXCR2, many of the ligands associated with those receptors, IL-1 alpha and IL-1 beta, again, not surprising, as well as other members of that family. And so, so this suggested that there was a good association between more bacteria and these chemotaxis factors, as well as IL-1 beta. The next thing that Camilla did was ask, what about neutrophils? And so what Camilla did was she took a cell estimation algorithm called Excel and was able to look at neutrophil abundance. And what's shown in the middle panel is the neutrophil abundance by this algorithm, Excel, with an Excel score. And what you can see is that there's an increase in the score from those biopsies from patients, suggesting an increased neutrophil abundance. She was then also able to show that that was associated with 16S abundance of bacteria, shown here in this particular plot. And she was also able to show that neutrophil abundance correlated with host IL-1 beta gene expression. So a lot of associations that you might expect to see between the skin microbiome and immune responses, both IL-1 beta as well as recruitment of neutrophils. So that's total abundance of bacteria. What are those bacteria? What particular bacteria are we talking about? And so this is the 16S data that was the 16S sequencing of the bacterial swabs. These are abundance plots where each line represents an individual patient. Each bar represents an individual patient. And what you can see is a couple different types of dysbiosis. You see a lot of patients, the dominant number of patients have Staphylococcus associated with their lesions, but you also have patients that have Corynebacterium, you have patients with Streptococcus, and you have patients that have a lot of anaerobes. And so that's what's happening in the patients as opposed to the contralateral skin of those individuals, which is heterogeneous. And so there is a dysbiosis that develops, and there are different types of dysbiosis. And so at the moment, what Camilla is working on is looking at specifically whether it matters what type of dysbiosis you have. And she's also able to now look at Staphylococcus, not just at the genus level, but also the species level, and begin to look at what staph is important here. Not surprising, you'll find that it's a lot of it's Staph aureus, and we're looking at different strains from the patients to see whether it matters which Staph aureus is involved in the infection. So that's ongoing, and I don't have much more to say about that, but I wanted to finish off by just asking the other question, and whether or not these particular bacteria have an influence on the immune response, and then subsequently whether they influence the outcome of the infection. And so to do that, we had to go back to a mouse model. And so these are experiments that are being done by a senior research investigator in the lab, Tej Singh. And so what Tej was able to do was take two different isolates of Staphylococcus, Staph aureus and Staph epidermidis. And these were isolated by Tori Lovins in the Grice lab from the isolates that we brought from Brazil. And what he did was associate these mice, B6 mice, without barrier disruption for 5 days, and then ask what happened in the skin. And so the first thing you'll see in the middle panel is that there's an increase in T cells compared to control skin with both Staph epi as well as Staph aureus. But interestingly, there's differences between what happens with Staph epi versus Staph aureus. And so this shows Ly6G as a marker of neutrophils versus Ly6C. And what you can see is that Staph epi induces recruitment of neutrophils, but Staph aureus induces a significantly increased number of neutrophils. And this is showing percentage, but it's also true in terms of numbers, whereas Ly6C positive cells don't really change. And so what might be driving that, what we think is driving that is Th17 cells. And so when Tej began to look at the T cell responses, if you isolated T cells from the skin and looked at their cytokine production, primarily in mice that had just been treated with PBS, there was a very small percentage of CD4 cells expressing IL-17. With Staph epi, there wasn't much of a change, but with Staph aureus, the particular Staph aureus that he's looking at, there was a significant increase in Th17 cells. And so finally, like, what does it matter in terms of the course of infection? And so again, doing the same type of experiment where he associates mice with either Staph aureus or Staph epidermidis, he then challenged these mice with Leishmania brasiliensis. And what's in the middle is showing you the course of infection, change in ear thickness over time. And what you see is that in red are those mice that have been associated with Staph aureus. In blue and black, you see either the controls or those associated with Staph epidermidis. So there's no change in parasite numbers. There's simply a change in the ear thickness associated with the infection. And correspondingly, when you looked at the 6-week time point when we sacrificed these mice, you see more IL-17 producing cells in those lesions. And so I think this is important because it not only shows that staph can be associated with increased pathology, but different staph may differ in their ability to induce increased pathology. And I will say, I don't have the data to show you today. We also now know that different strains of Staph aureus also have different abilities in terms of inducing this pathway, leading to increased pathology. So kind of to summarize the work on the microbiome, we know that in the infection, there is a dysbiosis that occurs. We know that these microbiota can promote increased IL-1 beta production, and that there's differences in both species and strains that influence the immune response in Leishmania lesions. We know that early on, there's an induction of ILC3s that contribute to IL-17 production, and Tej has a paper submitted looking at those ILC3s. We know that Th17s develop also a little later on after the ILC3s. A large number of chemokines that are produced leading to recruitment of neutrophils, which we think mediate the increased disease that we're observing, and also are a source of IL-1 beta, which can then amplify the severity of the disease. And so putting these two things together that I've talked about, we think there's multiple possible host-directed therapies for cutaneous leishmaniasis. I talked about those that might be blocking this pathway of CD8s, including tofacitinib, as well as NLRP3 inhibitors, anti-IL-1 beta or anakinra. But the dysbiosis also has some possible targets, certainly altering the microbiome by antibiotics or probiotic strains. And also we're looking now at whether or not we can block neutrophil recruitment as another possible way of blocking this pathway towards pathology. So the last thing I'll say, and it really harkens back to Paul Kay, his conversation or his talk yesterday, this raises the question of, okay, so we have these possible treatments, and the next step, of course, is taking them from a mouse model into patients. And so I think what Paul said yesterday is absolutely right. It's not easy. It takes a lot of effort. You need partners to be able to do that from companies. We've spent a little bit of time talking to companies about canakinumab, and scientists were really interested, but the administration, not so much. But our view right now is that we're going to continue to develop new targets as well as ways of blocking those targets, because my feeling is that we need as many possible ways to block this pathology because we're not really sure which ones are going to work. And so the thing we can do is continue to think of different ways of blocking the pathology. And one example of where it's really important is when you think about canakinumab. It would be wonderful to do a clinical trial with that in combination with antimony. However, it's probably not long-term from an economic point of view, very feasible from a long-term point of view. Canakinumab is very expensive. And so thinking about other ways you can block this pathway with cheaper drugs is probably important in the long term. And so I'll just end by reminding everybody that everything I presented was dependent upon the individuals on this slide. And with that, I will take any questions. Thank you.
Thank you very much, Phil. I think we are going to the next speaker, and at the end of the talks, we can discuss everybody together, okay?
Great.
So our next speaker is Lucas Carvalho. Lucas Carvalho is Associate Professor of Immunology at the Federal University of Bahia. He holds a bachelor degree in biological science and master degree in human pathology and PhD also in human pathology at the Federal University of Bahia. He completed his PhD in immunology at the University of Pennsylvania. He has experience in the field of immunology with emphasis on applied immunology, working mainly in leishmaniasis and also other infectious disease. So thank you very much, Lucas, for accepting our invitation, and we can share the screen. Okay.
Can you see my slides?
Yeah.
All right. So thanks, Cláudia, for the nice introduction. And thanks, Ebert and Daniel, for putting this excellent seminar together and for giving me the chance to show my data here today. So this is, I'm glad that Phil just gave a nice, background about the disease and as well about the importance of the new forms of therapies. And I'm going to talk today about a little bit about pathways that we can block, and as well as some candidates that we can probably use to decrease inflammatory response in cutaneous leishmaniasis. All right. So Phil just showed a little bit about this this clinical spectrum of the disease, and I'm just going to stress out a little about the immune response in these three clinical forms of Leishmania brasiliensis infection. So most of the individuals develop the cutaneous leishmaniasis, and 5% of them will develop the mucosal disease, and around 5% of people that has cutaneous leishmaniasis will develop a disseminated form of disease. You can see lots of acneiform lesions or ulcerated lesions spread through all the body. So I just need to say about the immune response in these patients that the disease doesn't happen because of lack of interferon gamma. If you biopsy, as you showed, the microarray and RNA-seq data, they do have interferon gamma, and this disease does not occur as well because of a skewed Th2 type of immune response. You don't see much of IL-4, IL-5, or IL-13 in the lesion of these individuals. But we see lots of inflammatory response, and data accumulated since, 10, 15 years ago by our group and other groups shows that these patients, they have lots of TNF-alpha being made at the lesion site. And actually, if you block TNF, as it's been done in the past by Paulo Machado and Edgar Carvalho, in these individuals, you can ameliorate the lesion and the time to heal. But the big problem is the lack of response to therapy in these individuals is quite big. And as well as if you see the patients in the early phase of the disease, right before the ulcer appear, we see that those individuals, they have more chance to fail therapy than when they come with the ulcerated lesion. So this is just an example of a patient that came on day 0 without a lesion, just without an ulcerated lesion, and by day 120, this lesion was inactive. So, and when we see the therapeutic failure rates in these individuals, we see that around 28% of patients with classical ulcer fail therapy. When they come in the early phase of the disease, we have about 75% of these individuals failing therapy. We don't understand completely what's going on, but for sure, in the early phase of the disease, we have more parasites than in the late phase of the disease. We believe that the treatment just releases lots of antigen, and the immune response, inflammatory immune response, would kick in very early and just contribute to the ulcer appearance. And actually, if you take biopsies from individuals with we call early cutaneous leishmaniasis, those individuals that does not have an ulcerated lesion yet. And if you take biopsies from early cutaneous leishmaniasis patients and let it go for 48 hours in a media, we're gonna see that they do lots of TNF, just like as a patient with the late phase of the disease or patients that had an ulcerated lesion. And this is healthy skin from healthy subjects. And you see TNF, we see lots of IL-1, IL-6, and I apologize, every time you see this weird thing here, it's just a incompatibility between Mac and PC. This is IL-1 beta, okay? So what I wanna show here is that very early in the disease, we see inflammatory response going on in cutaneous leishmaniasis patients, and those individuals are very hard, very likely to fail therapy. 75% of them fail therapy. So, what else? At least in Corte de Pedra, this area that we've been working, we see lots of atypical forms of cutaneous leishmaniasis. And this is being more common, this is being more frequent. And atypical forms like this lesion right here, where it starts one lesion, then start to spread, and or some vegetating lesions, some lesions that's actually cure in the middle, and they had what they call recidiva cutis, and some actually disfigured lesions, and some very infected lesions with secondary infection, bacteria going on, and lots of necrosis. So those atypical lesions are being very common in Corte de Pedra, and we see up to 80% of therapeutic failure in these individuals. So what are the drugs that we have in the market to treat tegumental leishmaniasis, at least in Brazil? The drug of choice of Brazilian governments is still pentavalent antimonials. And we see that early cutaneous leishmaniasis patients and the atypical forms individuals, they do not respond well to pentavalent antimony. There is another choice, liposomal amphotericin. They need to be, the extremely high cost would actually made impossible to treat patients with this drug. It's more, the efficacy is very big, actually very high, but the cost doesn't let us to treat patients with this drug. And now we have the mutafosine, that the therapeutic failure spans between 25 to 35%. So it's a better drug. It's a tablet, so you don't need injections. And although it's high cost, for example, last year in this endemic area, we have about 1,000 or 1,500 cases per year, and the government gave us less than 10%. In drugs enough to mutafosine enough to treat less than 10% of these individuals. So we think about adjuvant therapies, and it's been tested in the past. The pentoxifylline, that's a TNF inhibitor. It works very well for those severe cases of mucosal leishmaniasis, disseminated leishmaniasis, but it doesn't work for the case of regular cutaneous leishmaniasis. Roque Pacheco and Paulo Machado also tested the GM-CSF, topical GM-CSF and intralesional GM-CSF, that they saw a good efficacy, but of course, these recombinant cytokines, they come with a high cost. So just to know what we're going to target, we need to understand the pathways of inflammation. And what's going on in the tissue of these individuals. And Phil just gave a nice talk showing the role of IL-1 beta. I'm just gonna I'll show some data in human, in the lesion of human, showing protein, and as well as the role of the cytotoxicity in the tissue damage in these individuals. So Dario Zamboni group showed a long time ago that in inflammasome-derived IL-1 beta induce nitric oxide mediated resistance to leishmania. David Sachs group as well showed that the IL-1 beta that comes actually recruit neutrophils in a non-healing strains of Leishmania major. And our group showed that CD8s contribute to IL-1 beta production in mouse model. So the work done by Fernanda Novaes in 2014, this microarray, showed a few possible targets to block in terms of inflammatory response. And we see here the normal skin in green, most of them in green, and here the Leishmania skin. And we saw that those genes that involved in cytotoxicity, like granulizing enzymes and perforin, they are upregulated in Leishmania brasiliensis skin lesion when compared to normal skin. Same of those inflammasomes like IL-1 beta and NLRP3. And we just went ahead and asked if the lesion of these individuals would produce, cells from the lesion would produce IL-1 beta. And this is true, right? So cutaneous leishmaniasis patients, cells from the lesion, they produce high levels of IL-1 beta compared with healthy individuals. And we see that IL-1 beta is correlated with areas of necrosis. Just adding a little bit to the literature in terms of the deleterious role of this cytokine. When we see what kind of monocytes are the main source of IL-1 beta, and again we saw that the intermediate monocyte, those called inflammatory monocytes that have a high expression of MHC class 2, it's associated with damage in tissue damage in arthritis and other autoimmune disease. So those intermediate monocytes that's expressed CD14 and CD16 are the main source of TNF, and they are the main source of IL-1 beta as well, as we can see when compared to classical and non-classical subsets of monocytes. So another pathway of immunopathology has been studied a lot, and Cláudia Brodskyn's group, Olivia Bacelar's group, and Phil Scott's group have shown that there is a role for CD8 T cells and granzyme B in mediated tissue damage in cutaneous leishmaniasis. Some of this work were done in mouse, and some of them were done in human. And what we want to find out is what's going on in the lesion of these individuals, again, the site of infection. And we see here that the number of perforin positive cells positive correlates with the number of Leishmania brasiliensis mastocytosis in the lesion of these individuals. Again, number of perforin cells correlates with lesion size in cutaneous leishmaniasis individuals. What was surprising for us that the frequency of CD8s and NK cells were pretty much similar in the skin of these individuals, but when we see the main cells actually degranulating in the lesion of cutaneous leishmaniasis patients, we see that around CD8, 5% of CD8 cells are degranulating compared with 20% median of the NK cells degranulating. So nevertheless, those cells contribute to granzyme production in the lesion of these individuals. And we see, if you take the biopsy again, put that in media and wait for 48, 72 hours, we see lots of granzyme being produced in the lesion of cutaneous leishmaniasis patients when compared with health subjects. And again, granzyme B nicely correlates with the lesion size. So So what about this connection between CD8 cells, NK cells, cytotoxicity, and IL-1 beta production? So we decided to block granzyme B and see what happens with inflammatory cytokines in cutaneous leishmaniasis patients. And again, this is now PBMCs, peripheral blood mononuclear cells, stimulated with soluble leishmania antigen. We see nice production of TNF, interferon, and IL-1 beta. And when we block granzyme B, we decrease those cytokines in all of the patients studied. So in summary for this part, we see that NK cells as well as CD8 T cells, they produce lots of granzyme, and this extracellular granzyme contribute to activation of macrophage and TNF and IL-1 beta production, enhancing inflammatory response in cutaneous leishmaniasis patients. So we have a few targets right here, right now. So it's IL-1 beta by itself, TNF, and as well as this is extracellular granzyme, or if you can say cytotoxicity. And Phil just showed a little bit about the available drugs to block the deleterious effects of IL-1 beta. So there are Ilaris, that's a monoclonal antibody anti-IL-1, and Akira Kineret, that's a monoclonal antibody anti-IL-1 receptor. Of course, you don't want to treat patients with a monoclonal antibody. An injection of an Akira would cost in Brazil 40,000 reais. It's almost $8,000. So we're not talking about treating patients with a monoclonal antibody, but we need at least a proof of concept to say that this cytokine is really not good. So it's really causing damage. And if you block the cytokine, we can prevent relapse to treatment. But luckily, there is the glyburide, the glibenclamide, which turns out to inhibit NLRP3 and decrease IL-1 beta production. And what Augusto did was take biopsies of cutaneous leishmaniasis individuals and put together with glyburide, and he saw that after 48 hours, you see a nice decrease in the IL-1 beta production. Not only IL-1 beta, but glyburide can actually decrease TNF. And interestingly, it doesn't mess with IL-10 or IL-6, showing that this the effect of glyburide probably on NLRP3 would just affect IL-1 beta and TNF. So there are other pathways that leishmania would induce IL-6, another pro-inflammatory cytokine. Okay, what about other stages of immune response, inflammatory response, other stages or other pathways? It's not new for us that prostaglandin is required for inflammatory response. So actually, when you treat individuals with anti-inflammatory, what you're doing is just inhibiting those enzymes COX-1 or COX-2 and decreasing inflammatory response. And this is just one example that Lucan published a few years ago, showing that the production of IL-1 beta is dependent on endogenous prostaglandin E2. So we went ahead and just asked what's going on in terms of these lipids, the ability of these lipids to downregulate inflammatory response in cutaneous leishmaniasis. And just to remind you, so arachidonic acid, it's a lipid, right? It's a fatty acid, what turns to be omega-6. When it's metabolized, you see the production of prostaglandins, thromboxane, and prostaglandin E2 series. There are 4 receptors for prostaglandin E2, EP1, 2, 3, and 4. And what we see in leishmania is that at least 2 of them, the 2 and 4, here's prostaglandin receptor 4, prostaglandin itself, and prostaglandin receptor 2. It's actually upregulated in this cutaneous leishmaniasis lesions. So what about if we measure prostaglandin? So prostaglandin 2, again, if you take the biopsy, let it sit for a while, we're gonna see that cutaneous leishmaniasis patients, they have increased levels of prostaglandin 2 in biopsy in lesion. And again, prostaglandin 2 production nicely correlates, positively correlates with the I1 beta production in the lesion of the disease, infected macrophages, I'm sorry. So if you infect macrophages with Leishmania brasiliensis, you're gonna see production of prostaglandin 2, and it correlates with the levels of IL-1 beta. So what about therapeutic failures? Here we have healing time and individuals that had low expression of prostaglandin 2 and high expression of prostaglandin 2. And we see that the higher inflammatory environment these individuals have with high levels of prostaglandin 2, the longer they take to heal. So showing that this is actually a pathway it's involved in inflammation for sure, and it's actually contributing to a longer time to heal. So again, in 2014, with the microarray work, Novaes was able to find out several pathways of inflammation that were present in Leishmania brasiliensis lesion. This is not new, right? So antigen presentation, innate cytotoxicity, inflammasome, ROS production. But on the top of here, we can see that there are some downregulation compared with a normal skin of a lot of fatty acid metabolism genes. So those fatty acid metabolism genes, some of them can be involved in wound healing. And we decide to take a look closer what's going on with the polyunsaturated omega-6 and omega-3 fatty acids. I showed you that omega-6 is the same as arachidonic acid. So both of them are essential fatty acids. It means that we don't make those, we just get those from food, and they are metabolized through the peroxisome proliferator activated receptor gamma, or PPR gamma. PPR gamma happens to be a nuclear receptor for lipids. And this molecule is expressed in adipose tissue, colon, and expressed in macrophages. And the function of PPR gamma is to regulate fatty acid storage and glucose metabolism. So how does it work? Why people talk about ratio of omega-6 and omega-3? So if you have a high omega-6 concentration, low omega-3 concentration, what you're going to have is pro-inflammatory response with lots of prostaglandin and leukotriene series 4. Prostaglandin 2 and leukotriene series 4. So again, they go through the same kind of pathways. They use the same enzymes. So there is an enzymatic competition between omega-6 and omega-3. So the more omega-3 you have and the less omega-6 you have, you're going to have the opposite picture. So omega-3 will give production of the eicosapentaenoic acids, like they are prostaglandin series 3, leukotriene series 5, anti-inflammatory icosapentaenoid like resolvins, that's very important for wound healing, and anti-inflammatory protectins as well. So our main question was, if we give omega-3 to the cells, would they just, instead of doing the pro-inflammatory response, would they just decrease it and switch for a more regulatory response? And what happens with the parasites as well? And this is what I'm going to show right now. So this is what just said, omega-6, it's signaling through PPR gamma, giving, inducing lots of pro-inflammatory mediators. And our question was, what about if you give omega-3? Would we be able to decrease these inflammatory mediators and increase resolvins, for instance? To begin, the expression of PPR gamma in the lesion of cutaneous leishmaniasis patients they negatively correlate with inflammation. So here, the less PPR gamma expressed in the lesion of cutaneous leishmaniasis patient, the more TNF-alpha this individual will produce. It can go the other way around too. We know that TNF-alpha and IL-1 beta decrease the expression of PPR gamma, right? And PPR gamma signaling can decrease the expression of a TNF. And this is the same with the other inflammatory cytokines like IL-6 and IL-1 beta, right? There is this negative correlation between PPR gamma expression and the amounts of these cytokines. Same with the NF-kappa B, okay? So what if we give the omega-3 to the cells, what happens? So this is the response to soluble Leishmania antigen, and this is again peripheral blood mononuclear cells. And when we give soluble Leishmania antigen, of course, we see IL-6, IL-1 beta production, TNF, leukotriene before. And when we add omega-3, we call EPA and DHA, we decrease the production of the pro-inflammatory mediators, IL-6, IL-1 beta, and TNF. And surprisingly, we actually see an increase in the leukotriene by before. And this is dependent of the PPR gamma. The GW9662 is a PPR gamma inhibitor, specific inhibitor. And when we add this, we lose the effect that we saw in this slide, the previous slide. So SLA induced, but the EPA and DHA in red and blue cannot anymore, or both together actually, cannot anymore suppress or inhibit the production, decrease the production of IL-6, IL-1 beta, and TNF. And but the PPR gamma blockage does not affect at all the ability of EPA and DHA to increase the levels of leukotriene beta. And as important as the downregulation of inflammatory response is what they do with the parasites. So if you take macrophages infected with Leishmania brasiliensis, in this experiment we see 60% of the cells infected, and we start to give, we give once the EPA or DHA or both together. After 48 hours, we see, this is what we see in macrophage, human macrophage, right? They can control the infection in vitro. And we see that actually the presence of EPA and DHA or both together, we see more, even more evident effect of parasite killing at least 48 hours. This is more evident here in the number of pharmaceuticals per 100 cells when we add at 48 hours those EPA, DHA, or EPA, DHA, omega-3, we see this decrease in parasite counts. So, so in summary, omega-3, it signals through PPR gamma, inhibiting inflammatory cytokines, TNF, IL-6, and IL beta. And for another pathway that we don't know yet what's going on, it induces leukotriene before, and we believe this leukotriene before has been shown by Valeria Borges that leukotriene before actually increase parasite killing. What about drugs? Do you have any drugs that can actually bind to PPR gamma and decrease inflammatory response? And the answer is yes. There's pioglitazone. Pioglitazone is an anti-diabetic drug. It ameliorates hyperglycemia and insulin resistance in diabetes type 2. And this is a PPR gamma with anti-inflammatory properties. So we did the same kind of experiment and added this drug to peripheral blood mononuclear cells from cutaneous leishmaniasis patients, and we saw that in presence of pioglitazone, we saw decrease in TNF, IL-6, IL-1 beta, metalloproteinase 9 that we know that's been associated with tissue damage in cutaneous leishmaniasis, and as well as soluble CD163. Does not mess with IL-10. Again, when we block the PPR gamma, we completely lose the effect of the pioglitazone here in white. What about parasite killing? It does not mess with parasite killing at all. Actually, it seems that when you see the percentage of infected cells, there is no change. But when you see the number of mastocytosis per cells, we see that there is a slightly decrease in the number of parasites within macrophages in vitro after we add pioglitazone. And because this is 4 hours, maybe there is a lack, a decrease in internalization of parasite that goes on until like 24 hours. So in summary, what I showed today is that omega-3, EPA, and DHA, and as well as pioglitazone, they can be signaling through PPR gamma in macrophage, and you're able to decrease the inflammatory response in cutaneous leishmaniasis patients. We think this can be without affecting parasites survival, actually increasing parasite killing. And we believe in the near future we can do clinical trials with that. So the idea is not giving oral, it's just give a topic, like make a cream and treat as a adjuvant, put as a adjuvant with one of the drugs, miltefosine, or even the pentavalent antimony. So with that, I'd like to thank Daniela Celestino, was my student that did all the IL-1 beta experiments. All my colleagues from the endemic area, de Lagos, and Luiz Guimarães and Paulo Machado, the physicians that visit endemic areas. My colleagues from UPenn, Fernanda Novaes, Camila Morim, Dan Byron, Phil Scott, and Thais did all the cytotoxicity work. Mauricio Nascimento has been working with the omega-3 experiments, and Ravena with the pioglitazone experiments. And I would like to thank, and I think the questions are coming afterwards.
Thank you very much, Lucas, for the nice talk. Very important. And now it's a pleasure for me to introduce Danielle Walnis, who is one of the responsible for this amazing symposium. So Danielle is graduated in biological science at the Federal University of Ouro Preto, master's in biological science, biophysics at Federal University of Rio de Janeiro, and PhD in biological science, also at Federal University of Rio de Janeiro and Harvard Medical School. He did his postdoctoral in University College London, England, and now he is currently an associate professor at Federal University of Espírito Santo, and works on projects that address the impact of T and NK cell immunosenescence on the immunopathogenesis of human cutaneous leishmaniasis. In addition, it works on projects associated with differentiation of resident memory cells and immunoregulation during cutaneous leishmaniasis. Thank you very much, Danielle.
Thank you very much, Cláudia, for this introduction. Can you hear me? And also, can you see my screen?
Yeah.
Okay. Thank you, Phil. Thank you, Lucas, for accepting to participate of this meeting. It's amazing to share the space of this roundtable to discuss a little bit about immunopathogenesis. Also, I can confess that was amazing because also you guys made my work a little bit easier. Both described amazingly about immunopathogenesis, so I'm going to pass a bit fast about some details, and then I'm going to be focused to describe a little bit about immunosenescence. So before I move to the proper details about pathogenesis, I would like just to give a brief background about immune response against pathogens and cancer, which are we're going to discuss, and also which I'm gonna include senescence and senescent cells in these topics. So immunity against pathogens and cancer, and doesn't matter if it's leishmania or talking about pathogens, doesn't matter if leishmania, bacteria, fungus, or whatever. So a perfect frame or a perfect scenario related with protection is associated with a balance between immune activation and also immune regulation. When we have this is not going well, or when we have an exacerbated immune response or a decreased immune activation, we have in both cases disease association. So, but they are associated, or the disease are associated in two different ways. The first one, we have pathology, especially caused by the pathogens that grow easily in the body of the person. In the second size, we have an increased immune response, which also is going to cause pathology. And then in this, I'm going to be focused, which is the immune pathogenesis. So today, I'm gonna talk a little bit immunopathogenesis by using our study model, which is cutaneous leishmaniasis. Phil and Lucas also gave amazing introduction about the clinical spectrum of the disease. So we are interested to understand especially how the immune response, but the immune response also driven by senescent cells, which I'm going to tell you a little bit, is going to be associated with the pathogenesis of the cutaneous leishmaniasis. But when we look at the clinical spectrum of the disease, we can see clearly how the immune system are related with the disease per se, as Phil showed many times in his slides. We can see here, using the mucosal spectrum and also the localized clinical form, we have a strong cellular immunity associated when we can see very low parasite in lesions. So which means that the disease is much more driven by the immune system. So also, I'm not going to tell too much about the protection and the pathology or the elements related to this, but especially in pathology, Phil and Lucas showed many elements can drive the pathology, especially granzyme B, perforin, which means the cytotoxicity activity. As well, the inflammation is related with pathology. So today, I'm going to introduce a new element in this big soup. So I'm going to talk a little bit about senescent cells, which I'm going to tell you and I'm going to show you that is associated with the dark side of the pathology in cutaneous leishmaniasis, but also is associated with pathology of many other disease, not related only parasite, but also autoimmune disease or inflammatory disease. So I'm going to show you, or I'm going to spend a little time trying to convince you guys that senescent cells, or how senescent cells may cause pathology in different disease. I'm going to also tell you a little bit about some published data from our group. And also I'm going to show some unpublished data related to the whole of NK cells, T cells related to this pathology, to this immunopathology. And I'm going to finish my talk also with some unpublished data showing the whole of non-immune senescent cells in lesions of cutaneous patients that can magnify the immunopathology of the disease. So that's the thing that I'm gonna tell you a little bit. So before tell you about the proper data, I'd like to introduce some concepts. What is immunosenescence is an important concept that we must clarify. By definition, immunosenescence is a regular change that we get in our immune compartment. So normally, this change we get when we get aging, but also is well documented that this change also can occur by antigenic chronic stimulation and also during infections, which means that is both are very important and easy to induce changing this immune system. So this change can be observed in both compartments. So we can also look at in the innate immune compartment and the adaptive immune compartment, and this change can be phenotypically and also functional change, which gonna make in the host an impaired ability to respond to antigens. These antigens can be new antigens or old antigens, but also is associated with the severity to infections. But in the end, also, this change caused by immunosenescence imprint to the body a low grade of systemic inflammation, which we can call inflammaging, and this can easily be associated with aging and also during chronic infections. So moving to another concept that I would like to clarify is related to senescent cells, because during the immunosenescence, cells become senescent, and normally they have some features that can be found in all compartments, innate immune compartments, also adaptive immune compartments. So one features that I would like to talk about is the incapacity to proliferate, which is driven especially by the DNA damage that can be caused by proliferative, the excessive proliferation, but also can be induced by UV or cellular stress, which is going to activate several intracellular pathways, especially related to proteins that control the cell cycling, stopping the cell cycling, and also activating some kinases, especially I would like to call attention to the P38 MAP kinase, which also is involved with the second feature that I would like to talk about, which is the increased capacity to secrete pro-inflammatory contents. These pro-inflammatory can be cytokines, chemokines, growth factors, and proteases, which is associated with the persistent inflammatory environment, as I told you, the inflammaging, and also this content, this inflammatory content, can also driven senescent in a paracrine way, which senescent cells make more senescent cells. And this is quite interesting when we look at cutaneous lesions, which we have an increased inflammatory environment, and then we can have easily this phenomenon occur in the lesion. So this capacity to secrete the pro-inflammatory contents is called senescent-associated secretory phenotype. And also we can measure this to check some features of these senescent cells. So also senescent cells can be observed in the T cell compartment. So senescent T cells and also senescent NK cells share the same features that as I spoke previously. It's like a diminished proliferative capacity and increased capacity to secrete pro-inflammatory cytokines. But when we look into the memory compartment, we can see easily an accumulation of late differentiated memory populations, which we called TEMRAs. It's an effector memory that re-expressed the CD45 array. And these represent the late state of the differentiation of T cells. Also, senescent T cells has other features that I would like to talk about, also because we used some features to do the phenotyping of the senescence in cutaneous leishmaniasis. One is we can find easily in this senescent population a loss of expression of CD27 and CD28, an increased expression of CD57, which is a differentiation marker also used in NK cells. Also, these cells has shorter telomeres. They have diminished capacity of the proliferate the telomere by the telomerase. And also, I'd like to pay attention about this, especially the pronounced capacity to secrete and to make cytotoxicity. So, before I continue, I would like just to take a home message about this, the senescent feeders and senescent cells. Senescent cells accumulate in tissue and also can change the microenvironment of the tissue. So leukocytes, especially T cells, acquire new functions, especially related to cytotoxicity. Also, they develop new functions related to be prone to secrete pro-inflammatory cytokines, and these inducing this cell dysfunction also in neighborhood cells. And if we consider cytotoxicity and inflammation, we're going to have a perfect storm to create pathogenesis. This is already observed in rheumatoid arthritis, diabetes, lupus, COVID, I'm going to tell you a little bit later, but also cytotoxicity and inflammation are the main signatures of the cutaneous leishmaniasis immunopathogenesis. And then these arise the possibility of senescent cells helps this process and is making some whole related to this immunopathogenesis. So, I would like now start talking about specifically the whole of senescent cells, especially T cells and NK cells, in the contribution to pathology of the immunosenescence, or the cutaneous leishmaniasis, sorry. We're gonna start answer this question a couple of years ago. This work came up in 2019. Was conducted mainly by Luciana Covri. Luciana Covri is a postdoc in a Arnis laboratory at UCL, College London, and also developed some works here in Brazil, in my lab. So in this work, we used untreated patients diagnostic with cutaneous leishmaniasis caused by Leishmania brasiliensis. We used also health controls, which were age-matched, and also they came from the same endemic area. So before, we started doing this work by doing a huge phenotypic analysis in circulating T cells. We used CD4 cells and CD8 cells to do this analysis. And then we choose well-recognized market of senescent cells. We're starting doing this by using CD57, KLRG1, and phosphogamma A2AX, which is a marker for DNA damage. In all analyses, we could see that in both compartments, in CD4 and CD8 compartment, patient had much more expression of these markers comparing with health controls. So health controls are this is white box and cutaneous leishmaniasis as the black box. We can see here for the whole markers, the patients had more these senescent markers. So we also analyzed the telomere length for these T cells. So isolated CD4 and CD8 T cells from circulation by magnetic beads, and then we did the stainings for telomeres by using a probe, a fluorescent probe. So we could also measure that in patients the telomeres are shorter than controls. This was found in CD4s, but also CD8 T cells. We also saw an increase expression in P38 MAP kinase, which I told you before, that is a kinase associated with the senescence, which was very exciting. And then to check about the profile of the memory population, we used CD45 array and CD27 to discriminate between memory compartments. By using this, we can see here the different compartments from naive to emerald cells. And our analysis also show, especially in CD4, an accumulation of emerald cells in patients, which was not found in health controls. The same was found in CD4, but it's already known that CD4 get less senescent feeders than CD8 T cells. So our analysis also now in this specific population, taking emerald population and analyzing, so we could see that they proliferate less than emerald from health controls. This was found in both compartments, so in CD4s and also CD8. CD8 emeralds from patients had less telomerase activity comparing with controls. We can see here also. And then when we start analyzing the functional activity of these cells, we confirmed that emeralds from patients are much more prone to secrete the inflammatory and cytotoxicity compounds than health controls. So we can see here, so, by unstimulated condition, but also under stimulation condition with anti-CD3, these cells can produce much more TNF-alpha, interferon gamma, granzyme B, perforin, and also CD107 than health controls. Another interesting things that we can see here is naturally emeralds from patients are prone to secrete these. So we don't need to stimulate them to get the production. So they are already activated and they are producing this kind of contents, these pro-inflammatory contents than controls. So we start thinking about the whole of this population in the immune pathogenesis. These cells are circulating cells, so but doesn't matter if they produce or they are prone to produce this kind of cytotoxicity or inflammatory if they don't have ability to migrate to skin and cause disease. So we took information from previous published paper, amazing papers from Cláudia Brodskyn and from Alda da Cruz, from FIOCRUZ, which they did. They took cells from patients, CD8 cells and CD4, and they stimulated them with L. brasiliensis antigens to check the expression of cutaneous leukocyte antigen, is a CLA. We performed the same experiment, but instead we look at the total population, the whole population of CD4 and CD8. We look at the populations into, or the subsets into the memory compartment. So this data also showed to us that less differentiated population, but only high differentiated population like effectors T cells and also emerald cells have ability to migrate to skin, so which could be associated with the immunopathogenesis. So, and then we did a correlation with lesion size when that confirmed this hypothesis, and we found a strong correlation related to effectors and emerald, but we didn't see any correlations between naive CD8 T cells or central memory T cells. This correlation also was found in CD4 compartment, but I'm just gonna show in the CD8 T population. So another interesting thing is NK cells also can be senescent under the same situation, submitted to pro-inflammatory cytokines, and also a NK cell or a senescent NK cell has same features that senescent T cells, which means they acquire much more pro-inflammatory capacity, but also they express several senescent receptors, which can be tracked to determine the senescence in this population. So, and then we did the same analysis in senescent population of cutaneous leishmaniasis patients. This data was published in 2020, was also made mainly by Luciana, in which we start analyzing NK cells. This is a circulating NK cells. This analysis by gating CD56 population positive, we could see two different populations, NK bright and NK dim. NK dim here represents the late differentiated NK population. So as Luca shows previously, we didn't also see any difference between the frequency in terms of NK cells comparing controls and patients. But when we analyzed the state of the differentiation in NK cells, we could see an accumulation of late differentiated NK cells in patients comparing with controls. So also analyzing some senescent markers in this population, we confirmed this. So especially look at in the senescent subsets, we can see here an increase of CD57 receptor, an increase of KLRG1 expression, and also a decrease CD27 expression was not significant, but it's a tendency of to be less than health controls. And then we starting thinking, okay, we know that EMRA cells, EMRA T cells are really pro-inflammatory, and they participate of the pathogenesis by our analysis comparing their presence with lesion size. So we did the same, but also we decided to do a comparison between NK cells or senescent NK cells and senescent T cells to see which could be much more associated with the pathology of the disease. So I didn't mention, or I didn't show, but also we saw in the NK population short telomeres, low proliferative capacity, and also a pronounced pro-inflammatory profile. So, and then we started doing this analysis comparing some functional capacity between senescent T cells and senescent NK cells, which shows us that in circulation, senescent NK cells from patients are more prone to make cytotoxicity, which is associated with much more granzyme, perforin, expression of CD107 comparing with EMRA from patients. Is the same also, they proliferate much more than EMRAs from patients. But an interesting thing is senescent NK cells, they didn't express too much CLA, which means that they don't have too much capacity to go to skin as CD8 T cells used to do. So to analyze this, we took biopsies from patients, and we stained for NK cells, we stained for CD8 T cells too. Also in this stain, we associated granzyme B marker and also CD57, which is a senescent marker. So in our analysis, we could see that we could see both population in skin. So, but we could see much more an association of CD8 in skin than NK cells comparing. And also when we did a correlation, we could see that CD8 T cells could correlate with lesion size, but in our hands, the frequency of NK cells were not correlated with the lesion size. On the other hand, when we analyzed the senescent population with cytotoxic capacity, we could see similar data in terms of the accumulation of skin, but again, senescent CD8 accumulates much more than NK cells, than senescent NK in skin, but a correlation changed. We could keep seeing the correlation between senescent CD8 cells, but also now we got a correlation, was quite weak correlation, but was significantly related to senescent NK cells too, which means that senescent population can do some different works than non-senescent population into the skin. This data also was published. It's a mix between the data was published by Luciana and also Renan Garcia is a doctor, PhD student from my group. So we confirmed this. This is unpublished data that also we are working on it now. When we got directly biopsies, skin biopsies, and also health control skin, we did enzymatic digestion, and then we did flow cytometry with the cell isolated, or also we did a sorter isolation to do functional experiment with NK population, CD8 population, and CD4 population. I'm not going to show too much related to this. I'm going to be focused in CD8 T cells that going to confirm the things that we showed by immunofluorescence. So this is the gate strategy in skin. So I'm just going to show, because it's quite hard to gate proper the cells after isolation. We have a lot of debris. So and this is the memory profile that we got from this population isolated from skin. So we can see here MRAS population, effector population, naive and central memory. Comparing with patients, skin has a different profile than blood as expected. But one thing that we didn't expect it, to be fair, we thought that we could see much more MRAS than in skin than in blood, which was not happened. In skin, we can still see a lot of MRAS cells, but also is much more effector cells than in skin, than in blood, sorry. So our analysis related to CD57 expression and granzyme expression shows things that we thought before. So CD57 is related with the late differentiated population, and also the granzyme B capacity is also associated with the both population or late population in lesions and also in blood, but we didn't see too much related with naive or central memory populations. So which confirms our data before. Now I would like just to move to new data that was recently accepted for publication that confirms part of the data showed previously related to senescent signature in cutaneous leishmaniasis lesions. So this data was done by Carlos. Carlos is a PhD student from my lab. This data is already accepted in immunology. I think is going to come up very, very soon. Carlos took the data set deposed from Scott group. He analyzed the data from this paper, the Amorim 2019. Carlos' analysis shows around 2,000 expressed genes, between 6,000 upregulated and 6,000 downregulated genes. Ontogeny analysis in those genes showed that they are associated with the different cell functions. I would like to pay attention, sorry, in aging and also replicative senescent, which we could find around 195 genes, around 115 up and 79 genes related to those functions downregulated. Analysis in these genes taking key senescent genes shows that most of the important senescent genes, which is P21, P16, P38, MAP kinase, SES32, and others, were upregulated in lesions comparing with health skin. We could see, I'm not going to show here, but we could see some correlations between the parasite number found in lesions. But an interesting thing is here, we didn't see correlation between senescent genes and age of patients, which means that the expression was driven by immune response or parasite stimulation and not related to the age patients as senescent genes used to be normally in a health condition. So another thing is the convolution analysis of the cells that could be in potential presenting lesions shows that those genes, the senescent genes, were much more associated with late differentiated population in T cell compartments, as I spoke. It's like EMRA cells and also effector cells. And also we can include senescent or late differentiated NK population. Our staining, just to confirm this, we did a staining for KLRG1 and ATF in lesions by immunofluorescence, and we could see an increased expression of those markers, which also is similar to the gene expression. And then we also found a correlation between the expression of those senescent markers with lesion size, which means that senescent genes really are associated with lesion size and severity of the disease. So then we moved to the analysis specifically related to the signature with the immunopathology, and then trying to connect the immunopathology signature with the senescent cells. So the first thing that we analyzed was the SASP contents, is that a secretory phenotype associated to senescent cells, and also inflammatory immune response. So here we can see the profile that we found. We have here chemokines and cytokines, and also we have here SASP. Comparing health controls with patients, we could see a completely different profile in terms of genes expression. So here's the whole profile of the genes analysis, and our deconvolution also shows that most of the SASP genes and inflammatory genes are related with senescent population or late differentiated populated and non-other or less differentiated population. In a similar way, we start to look at the cytotoxic genes, which also is very well known. And then the interesting thing is most of the deconvolution correlated those genes or the genes related to cytotoxicity, which could be receptors or contents or intracellular contents with senescent population. So this suggested us that the cytotoxicity in lesions and also the pathology is driven by differentiated population, which includes the late differentiated population, and suggesting that the senescent cells could in this compartment could be or could cause or help with the pathology. So I'm moving for the end of the the presentation, and I would like just to show a brief data or a bit flavor of the things that we are doing, also related with the non-immunosenescent cells in cutaneous leishmaniasis, in which we are trying to see if those cells can also help the pathology direct or in an indirect way. So when we look at the skin, we have several different cell types there. We can see immune cells or cells from immune compartment like resident T cells, dendritic cells, resident macrophages, but also we have non-immune cells or cells that is not considered immune cells like keratinocytes, melanocytes, and one special cells that calls our attention is the fibroblast that I'm gonna show you a little bit. Fibroblast is a really important cells in the research to understand the senescence, especially that came up from the 60s from Phillip. The replicative senescence was discovered in studies by using fibroblasts. So senescent fibroblasts do not proliferate, but also they are really well able to secrete SASP. Also, they have all the DNA damage markers that I spoke. And also, these cells are related to the pathogenic role in several diseases, skin disease and non-skin disease like sclerosis, systemic, psoriasis, skin cancer, which could also suggest us that they could drive or help to understand a little bit the immunopathogenesis in the skin. So I told this before, but also a SASP produced by fibroblasts can induce the immune recruitment to the skin, but also can induce a senescent in a paracular way, so which could help in this environment. We have late differentiated population and senescent population. So we start analyzing this with the same experiment. We took biopsies from patients, and also we took biopsies from health controls. We isolated fibroblasts in early passage at the 3 or the 3rd passage maximum that we used. And then the first thing that we did was we stained the fibroblast isolated from health controls and isolated from lesions for galactosidase associated with senescent cells, which is called SA beta-gal. We could see much more the fibroblasts stained in lesions for beta-gal than health controls. This was confirmed also in all our experiment. And also we analyzed DNA damage marker in those cells, which also confirmed that the fibroblasts that came up from lesions have already a senescent profile comparing with fibroblasts from non-regular lesions. Now, or non-lesional lesions, sorry. Now we are doing some co-culture experiment trying to see if these lesional fibroblasts are able to induce senescence, and also we are doing a pan-phenotyping of these cells, trying to understand a little bit more the whole of this cell in the immunopathogenesis. So just showing this amazing slide from review from Novaes and Phil that came up a couple years ago. Phil, permit me introduce a little bit some components here. So the thing that we are thinking now in our hypothesis is we have these tons of elements driving pathogenesis in the cutaneous leishmaniasis. The things that we are trying to add here is that senescent cells, senescent T cells and senescent NK cells can also cause pathology, helping the inflammatory process and also helping the killing directly. But also we are trying to see if the senescent cells that could be driven by this inflammatory process could enhance this process or these mechanisms that we can see during the disease. So to conclude, I would like to take some notes about that. Our data shows that the circulating and lesional senescent cells accumulate during cutaneous leishmaniasis. So these cells have increased secretory phenotyping. Also, they have a lot of cytotoxic activity, which can help the immunopathogenesis. Also, we are showing that no immunosenescent cells may enhance the lesional mechanisms related to immunopathogenesis, and these together arise another thing that Phil discussed previously, but also add that the blocking senescent cells or blocking senescent mechanism may offer a new possibility for therapeutic targets. So that could be cool, and it's one thing that we are working now, especially related to P38, which was by using a P38 inhibitor, which was already used in humans, that we are doing to help us understand the senescence and the pathogenesis. So just to finish, I would like to thank some people from my university. First of all, I would like to thank the students mainly that make this work. Renan Garcia, Carlos Fanteselli e Dr. Luciana Coveri, Renan Garcia and Carlos Fanteselli here. Also people from our lab that support us most of the time. Some collaborators from my university that help us, especially in the pathology stains process, especially Sandra Venturin. Also would like to say thank you to Professor Aloysio Falqueto, who is our hand in the clinics and is helping us for a long time in this project. Also, I would like to thank in special Professor Arne Akbar in some members, his member group, that also helped us a lot in this project. Professor David Moser, Professor Phillip Scott and Camila Morim for collaborations, especially in the RNA-seq data. I would like also to say thank you to Herbert and Cláudia Brodskyn from FIOCRUZ in the collaboration with the Senescence, and Ana Caetano for Senescence studies too, especially related to COVID-19 that we are taking in place. So thank you so much for everybody.
Thank you so much for your nice talk. And now we are going to start our discussion. We have already some questions here, but if there are someone that would like to ask a question, please feel free to do that. Herberty, some questions?
Are you going to start for the chat or for us, Cláudia? Sorry.
I think that okay, I can start from the chat. So the first question is, question is there I think it's to Phil. Is there a correlation of disease progression and I1 beta production by inflammation other than NALP3?
Yeah, so the other inflammasome that so again, one has to keep in mind we're only looking at gene expression, and so that's an important distinction in what we can see. And I think actually I can comment later about the fact I think that protein expression and what Lucas and Dan presented suggests you can find even more when you look at protein. But in terms of gene expression, the other inflammasome member that we saw highly upregulated was AIM2, which is often associated with NLRP3.
Yeah, I also observed this expression, higher expression of AIM2.
Yeah.
Some of the Luis Afonso. Phil, given that NALP3 activation and I1 beta production are dependent on P2X7 receptor activation, have you considered evaluating gain of function mutations in P2X7 in patients with increased pathology?
Yeah, Luis, that's a great question, and so we've spent a fair amount of time looking at P2X7 receptor as a mediator of inflammasome activation in the mouse models, and unfortunately, it hasn't been particularly successful. And so I'm not convinced that, at least in the mouse models, it's playing a huge role. But because there are these, patients or individuals that have changes in P2X7 receptor functionality, it might be worth looking at. We would have been really interested in looking if we had some data suggesting it was really important, but so far we haven't gotten that.
Okay. So now there is one from Luciana Covre. Thanks, all amazing presentations. Dr. Phillip, is there an association between cytokines, I1 beta and others, and a non-specific response as observed in NKG2D mice model or in lesions?
So I'm not exactly sure what the question here is, but
that non-specific response when you use LCMV?
Yeah, so if the question is, do we see IL-1 beta? So in that non-specific CD8s that are killing via NKG2D, that does lead to NLRP3 and IL-1 beta production. So that's true. In terms of other non-specific, other than CD8 responses, clearly in the dysbiosis, we see a rapid increase in IL-1 beta early on, which is probably non-specific as well, but we don't know that for sure.
Okay. Now we have a questions from Luciana Covre to Lucas. Very nice presentation, thank you. Investigating the role of PGA2 in the lesion, do you have an idea of how it can modify epidermal cells to impact in the healing process?
Right, yeah, this is a really good question, and we haven't addressed any experiments in the lesion yet, but definitely those, if we can in vitro turn these cells into a more anti-inflammatory cells, we believe that we can do this in vivo as well, but we haven't addressed any clinical studies with that yet.
But I think there is a kind of receptor specificity, no? Of when we think about PGA2, because PGA2 also could be really inhibitory response, not so inflammatory response.
That's true.
When you have the balance between PGA2 and LTB4. So I think depends on the EP2 and EP4. What do you think?
Exactly. There are some papers that says that the PGA2 can act as a anti-inflammatory as well. So it depends on the receptor, as you said, Cláudia, and we have to tease this apart and see what's the receptor in this case. In the case of leishmaniasis, of leishmania, what actually is the receptor that is turning this response into pro-inflammatory, and try to hit that.
Okay. One question from Camila Farias Amorim. I don't know if I missed this information. I think it's to Danielle. Did you look at the lifespan of senescent CD8s compared to non-senescent?
Yes, Camila. I didn't show this data, the senescent are more prone to apoptosis than non-senescent cells.
Mm-hmm. So could create an anti-inflammatory, in this case, an anti-inflammatory environment, do you think? No, because it's really pro-inflammatory, or because they liberate the septs before?
I don't know, Cláudia, that's a good question. The senescent cells are amazingly pro-inflammatory. So, and I mean, effector cells, when I say senescent, I'm telling about humoral cells, not effector, but both populations are amazingly pro-inflammatory. Talking about the ability to be apoptotic, humoras are much more prone, even comparing with humoras, with effector cells, which I think they are more susceptible to dying lesions than effectors. I don't know if I answered the question.
I have a doubt, because I know that you have specific markers for senescent cells, but also you have PD-1, no, in these cells, the expression of PD-1 could be.
Yes, yes, but PD-1 is much more expressed in effectors than humoras.
Okay.
So when we look the expression of most of the exhaustion markers, like a PD-1, LAG3, TIM3, CTLA4, in our hands, we saw much more associated with effectors than humoras. So,
but these effectors could be changed to humoras. There is a kind of transition and could be an exhaustion before senescence, or?
Yeah, I think it's the step before become humoras.
Yeah.
Yeah, it's a step before humoras.
All right. Dario Zamboni, congrats on 3 great talks. To Phil, I wonder if you already detected ESC Ponta using the ESC C3 in intermediate monocyte, as Lucas shown, as a proof of principle that this system works?
Yeah, that's a great question. No, we haven't done that yet. We're still looking at the individual populations. So so no, but that would be a good thing to do. We haven't done it yet, Dario.
Okay, the second question. It was previously shown that ESC Ponta can be liberated during pyroptosis, and of course be phagocytized by other cells. Did you guys consider this cavity?
Yeah, so we also aware of that. And so where we're looking at that right now is in vitro experiments. It'll be a little more difficult to do that in vivo, but we are looking at in vitro at whether or not the specs are being released.
Okay, so these are the questions here in the chat. If you guys have questions, please.
Yeah, good question.
We'll have Nathan and Herbert. Nathan, please.
Yes,
Nathan,
so a question. Thanks, Herbert. A question for Phil and Daniel. I'm wondering if you know what's recruiting, and perhaps more importantly, what's retaining these bystander CD8s in the lesions. Dan, you talked a little bit about CLA. So I ask because we often think about recruitment as being fairly non-specific, but retention is heavily influenced by TCR engagement. And so in terms of that specific population, is there anything that is specific to that pathological CD8 population, or just recruitment and retention it's too broad, it's too redundant, and there's no hope of preventing having these CD8s come into these lesions in the first place?
Go ahead, Dan.
So that's a good point. One thing that we are checking now is the difference between the recognition or the antigen recognition between effectors and humoras. to see if they work in a different way, as you say. Also, I'd like to ask one thing to Phil after that. But anyway, yeah, I think they work in a different way in lesions, especially in terms of functionality. I don't think that they are different. I think the effectors and emeras, they work in the similar way. But the mechanisms by which they start working is quite different that we are believing now. Phil has amazing data showing the NKD2D, the cytotoxicity driven by NKD2D expression after infection with LCMV. So CMV also is well known to induce senescence in humans. So which we think that part of this bystander cytotoxicity is made by senescent cells proper, much more senescent than effector cells. So we are trying to check, especially the TCR machinery between those populations to see about that.
Yeah, and Nathan, I'll add that so some cells will go in and stay independent of antigen. So it doesn't require antigen for cells to stay, and that's what happens with resident memory cells frequently. But I don't think we know whether or not there's additional signals that are independent of specificity that are keeping them there. We have been really interested in the migration in, and again, whether or not one could block that. And in the studies that Camilla did, what she was able to see is that some of the genes that were associated with treatment failure turned out to be CCL3 and CCL4. And so we've been looking specifically at CCR5 and whether or not that's important. Again, we've been looking in the experimental models, not in the patients yet. And so CCR5, we think might be playing a role. Of course, the other cytokines or chemokines that are upregulated, very highly upregulated, are CXCL9, CXCL10, CXCL11. Not associated with treatment failure, but that doesn't mean they're not important. And so CXCR3 is probably going to be pretty important too, which wouldn't be a big surprise to anybody.
Right.
Herbert?
Thank you, Cláudia. I'd like to thank you all three. It was an amazing conferences. Phil, I have a question for you first. Congrats again for your talk. What about thinking when you use the model, so you put only CD8 cells, you have pathology, you put CD4 and CD8, you don't have it. So my question is thinking about the patients. Maybe the problem is not CD8 cells, but maybe CD40 cells that are not doing regulatory functions.
Yeah.
So do you think about that? Did you try to see something in the patients? But
yes.
Maybe CD8 is consequence, not the cause. So, sorry.
Yeah, so that's a very good point. And so experimentally, Fernanda did some experiments with Tregs, and I'm not sure the results were certainly clearly indicated. What you're thinking is that maybe there's an absence of Tregs there, so you're not controlling the CD8 response. That's certainly possible. We really haven't addressed that at all in the patients, and not too much in the experimental models. I will say that the other thing, and I should have said this, I mean, Lucas showed pretty clearly that it's not just CD8s, that NK cells are going to be playing a role there as well. But yeah, it's a good question, Herbert.
And my second question is about inflammation. That's for you and for Lucas. I think that's a question about the tolerance and the resistance. So maybe inflammation is important to control parasites, but at the same time as you get the pathology. So it's a balance. What do you think about that? So what's the impact of inflammation to control the parasites in patients? So maybe we don't have this answer yet, but maybe it's important. So the patient is trying to control the parasites and use the inflammation. But when you use the inflammation, you have the pathology. So it's a...
Yeah, so what we know from the models that we've looked at is that IL-1 beta and the inflammasome are not involved in control of Leishmania major or Leishmania brasiliensis in our experimental models.
Okay.
Dario has data, which you clearly are probably aware of, that with leishmaniasis, IL-1 beta may play a role in, does play a role in some control of the parasites. However, even in those situations with Amazonensis, the animals never resolve the infection. So IL-1 beta is pretty interesting, and it's clear that there are certain situations where early on, particularly, there's experiments that were done a number of years ago, that IL-1 beta will be important for developing a protective response. My view about this at the moment, to kind of put things together, is it's the amount that may be important. So just like TNF, a little bit's good, a lot's bad, and that may be true with IL-1 beta. Dario may want to comment on that as well. I think I see him on the.
Lucas, do you have something to add?
You are muted, Lucas.
Just getting back to your first question, we have this discrepancy between the peripheral blood and lesions. So if you take cells from peripheral blood from patients, you can easily downregulate inflammatory response with IL-10. But in the lesion, we see a fair amount of IL-10. I don't know where it comes from, if it's CD40 cells or other cells. But the fact is that we see this fair amount of IL-10, but we don't see, even if we add more IL-10, we don't see ability of downregulating inflammatory response in these individuals. Getting back from for the IL-1 beta, in our experiments, if you block IL-1 beta, I'm not sure about inflammasome in LP3, but in our experiment with human cells, if you block IL-1 beta, you don't see any difference in parasite killing. So at least the IL-1 beta is not necessary for Leishmania brasiliensis killing in human, but I'm not sure about inflammasome.
Danielle, do you have a question to Phil?
Oh yeah, I'd like to ask to Phil. Phil, you showed a slide, a very interesting slide about the MICB receptor expression in myeloid cells. Do you think that those cells must be infected to express MICB, or just from the environment they naturally, inflammatory naturally, they express these?
Yeah, so first, what I showed there was from patients, and it wasn't specifically in any cell type. But in the mouse model, when we looked at expression of NKG2D ligands, Ray1 alpha in particular, it doesn't have to be infected cells. And that's pretty well known. I mean, the inflammation often leads to upregulation of those ligands. So in lesions with Leishmania, at least in the mouse, you'll see a lot of the myeloid cells expressing Ray1 alpha.
Okay.
I think that you has a question. Hello.
So no, I just wanna comment a few. Hi, Cláudia. Good to see you guys. Pleasure to be here. So I totally agree with you. I think it will be very interesting to understand what's maybe a dose concentration or specific cell that are producing a lot more, or whatever is going on. It's very interesting, and I think that happens a lot of cases, right? So maybe in neutrophils, like we have some experiments going on with neutrophils. So a little bit is good, or maybe the opposite. So if you have a lot more, it's completely the opposite. And so I think in terms of the inflammation, I think it would be this is why I was asking these questions to Phil about the experiments to detect the in vivo, which is the really cells that are producing IL-1 beta and activating the inflammasome, because it's possible that some cells produce a little bit of inflammasome and then produces some IL-1, which is of course going to recruit another cells and neutrophils, and then perhaps neutrophils makes everything gets worse. Then as Cláudia mentioned and published, it's going to be the activation of the CD8 cells, and then it's going to like kill the infected cells and trigger more inflammasome activation. Then the whole thing gets out of control, and then of course it's detrimental and increased pathology. So I totally agree with this model. We should go to figure out when exactly the first cells get activated, the inflammasome gets activated in those ones, and then which cells are recruited to the sites, and then eventually why it is not ill. Then the inflammation continues, and then of course you may have a detrimental role of the inflammasome and IL-1 beta, of course.
Okay. Okay, I think Everett has, do you want to comment something?
I want a question for Lucas about
No,
the
about,
nutrition.
just asking if Phil, Daniel, or Lucas want to comment something about that use comment or suggestion. Or...
We agree.
Okay,
Me
Everett,
too.
please.
Lucas, did you look at the nutritional factors of the patients from Cós de Pedra? I don't know. They have, do they have a deficiency on omega-3 or omega-6s? So did you eat fish? Do you know that?
Yeah, we haven't looked the nutrition factors for those patients, but for omega-3, yeah, there was no actually difference between patients and healthy subjects. So why, it does not get into the lesion, something like this, to modulate immune response? I don't know, but the fact is, if we add more, right, to the cells, we can downregulate inflammatory response. We haven't looked, but this is the only thing that we looked in terms of nutrition factors.
I was thinking about resveratrol. If it is not here today, but to put the people to drink wine to see what's the effect, but the amount of wine to have enough resveratrol. I think Danielle worked with resveratrol too. It's a huge amount of resveratrol, but it's something to think about, to do over vitamins, omega-3, omega-6s, and to see what's the impact of these immune nutrients on diets of those patients. I think it would be something very interesting.
Definitely, I agree.
But in relation to the regulatory mechanism that you commented about, don't you think that is a problem of kinetics? So they produced IL-10, I think, in really a late phase when you have an inflammatory response that is very hard to control, or TGF-beta or whatever. And the amount of the pro-inflammatory cytokines is really hard to regulate. And you have all these senescent cells that I think I have already done this question to Daniel, but I am thinking if, for instance, if you can phenotype our cells and you can find some of these senescent cells circulating more than the normal, do you think that we are more prone to develop a more severe disease, or it's only induced by the infection?
Yes, so Cláudia, I agree with you. It's hard to modulate some when you have the cells in that in that end stage, they are already inflammatory. But we see this inflammation very early before the lesion appears, actually. So if you take the biopsies and put it in culture, you're going to see lots of cytotoxicity. Actually, granzyme B, we see even more in the early phase of the disease than in the late phase of the disease. Well, the number of cells in the early phase of the disease is, of course, lower, right? But we already see this high inflammatory environment. So maybe it's just too late to try to modulate immune response with a kind of regulatory T cells or something dependent on IL-10 production. So that's why I think reprogramming macrophages, this is something that's doable, right? You can take a macrophage that's inflammatory and then reprogram it to become a more anti-inflammatory. Then you get back with this point that Nathan showed yesterday.
Yeah,
We wanted to like a M2 macrophage that's going to allow parasite to multiplicate, and then you don't want inflammatory as well, because it's going to bring more cells in that's going to be infected. So we don't know what to do, right? So not too much, not too little. So
yeah.
Just to complement the things that Lucas says, this inflammation, and doesn't matter if at the beginning of the disease or in the late phase, will be very important to drive senescence. So you guys showed several data about the TNF, about the IL-1 beta, and those cytokines are amazingly to induce senescence in senescent T cells or NK cells, or even in non-immune cells. So which is, I think, as we used to see in leishmania, and we don't see in our patients, we don't see too much parasite. And doesn't matter the parasite there. The things that is feeding the senescence mechanism is inflammation. And this inflammation, of course, came up from the infection at the beginning, but I think also the DNA or the damage that is caused after the parasite be cleaned, that's going to keep the environment, the inflammatory environment, and also is going to keep the senescence there.
Okay. So I think we don't have more
I
questions.
have one question for Danielle. Sorry, Cláudia.
No, it's fine. It's fine. We have time.
Danielle, about the senescence, congrats for your talk, it was very nice. And I was wondering about the mucosal leishmaniasis, because Marcia from FIOCRUZ told me that usually more common mucosal patients when they are becoming old. So it's associated to aging. Lucas and Phil can talk about that. And I know that you talk about immunosenescence, but what's the impact of the senescence? So in the agents of maybe for this process for help it to spreading, it to become more inflammatory in other sites. What do you think about that, Danielle?
Yeah, that's a good point. Comparison, we have some mucosal patients here and also some experiments doing similar analysis in those patients. And I can tell you, in mucosal, we can see much more senescence feeders in T cell, in the T cell compartment, in the NK cell compartment, than cutaneous leishmaniasis, even when we age matted these patients. So, which means like much more inflammatory disease, we have much more senescence linked there. So between young patients and old patients, so also we can see much more senescence feeders in old patients than young patients. But I think now is because it's accumulation between senescent induced by the inflammatory process plus senescent that can occur naturally in those patients, right? Did I answer your question? You are muted.
It's not good to be old. It's part of life.
Come on. It's amazing to be old.
To figure out right now.
At least you survive, right?
To be old. Okay, I think we don't have more questions from the public, from the audience. So I would like to thank again the speakers, Phil, Danielle, and Lucas. Also, Nathan, Paul, and Dario, Herbert, we are here, and all the audience for this section. Thank you very much, and until tomorrow at the same time, no? 1 o'clock.
Yes.
Thank you.
Thank you so much.
Very much.
Hopefully we'll see in the next year, no, Phil?
Any word, Leish, for sure.
I hope so, Claudia. I hope so. I need a caipirinha on the beach at Porto de Bar.
Yeah, I can imagine. Me too, because you don't have caipirinhas in the beach also. So. Not only you.